Hi everyone, and welcome again to another video of our class Biology 2, and this time around we'll be talking about the second important topic in the central dogma of molecular biology. So first of all, last time we discussed about replication, but this time around we'll be talking about transcription and the accompanying post-transcriptional processes. Now, of course, we have to recall what would be the central dogma of molecular biology. So looking at this figure again, we now know that there are three important parts. This structure, of course, is called DNA, and the middle structure is called the RNA, and the RNA will be producing what we call as the protein or the polypeptide. Now, in addition to knowing that the DNA produces the RNA and the RNA produces the protein, you also need to know the different names of the processes. So when DNA produces another DNA, you know that this is called replication, while DNA producing the RNA is called transcription, and the RNA producing protein will be called as translation. And of course, by the end of our course, we'll be talking about a very special transfer wherein the RNA produces back the DNA, and this is what we call as reverse transcription. Since we're done with the DNA replication, we proceed now with the second important topic, which is transcription, where the DNA produces what we call as the RNA. And with that, our objectives for today's discussion is for you to, number one, explain the process of transcription from recognition of promoter region up to the termination, from the start of transcription up to the finish of transcription. And number two, for us to infer the importance of post-transcriptional processing, which implies that after the RNA is produced, certain processes still happen, at least for the eukaryotic transcription. So we'll talk about that a little bit later. So first, what would be the definition of transcription? What is transcription? Now, you're very familiar that transcription is defined as this. Take note that there are three important parts in this definition. The first would be the process of copying a segment of DNA nucleotide sequence. So the first one is the presence of a gene that is being copied, a DNA segment that is being copied. By number two, it is being copied by the enzyme RNA polymerase. Number three, into an RNA nucleotide sequence. So we can see here the central dogma all over again. So the gene, the DNA, is now being copied by the RNA polymerase into an RNA sequence or an RNA nucleotide sequence. So that's a definition of transcription. Now, looking at this figure, this will give us an overview of what transcription is all about. So you have here the double-stranded DNA, one of which would be the DNA template strand that will be used to produce your mRNA via the process transcription. So you will be producing over here the mRNA, wherein the different DNA nucleotides are base paired to RNA nucleotides to produce your mRNA. So we'll notice here that the A is base paired with U, the C is base paired with G, the C is base paired with G, so on and so forth. So that is the process transcription. Let's talk about that in detail in the next few slides. Translation, we'll talk about that after the few more meetings from now. Alright, so we need to make a comparison as well. So there is a great difference between the transcription of prokaryotes and transcription of eukaryotes. And take note that the transcription in the prokaryotes would be simpler compared to the transcription of the eukaryotes. Why is that? Because you can see here that in prokaryotic transcription, after producing the mRNA, it goes straight into the cytoplasm for translation. But for the eukaryotic transcription, there is an added step here. Inside of the nucleus, after the DNA has been transcribed to produce the RNA, take note it is called the pre-mRNA because the extra step is called the RNA processing, wherein the pre-mRNA would be processed to produce your mature RNA, which will then go out of the nucleus and then translated in the cytoplasm by the ribosome. So again, the transcription in prokaryotes would be will be simpler compared to the more complicated eukaryotic transcription. So let's talk about the different actors of the transcription. Let's look first at the comparison between DNA polymerase, which is part of replication, and the RNA polymerase, which would be part of the transcription process. So what's their difference? Particularly, they will have these three important differences. Number one, RNA polymerase will be catalyzing the linkage of ribonucleotides. So in this case, you will be making or in order for you to produce the mRNA, or the RNA, you will be making use of the adenine, the cytosine, the guanine, and instead of thymine, you make use of the uracil. So that would be a great difference between the RNA polymerase and DNA polymerase. For the DNA polymerase, it catalyzes linkage of the deoxyribonucleotides, which would include the adenine, the cytosine, the guanine, and then instead of uracil, you make use of the thymine. So this will imply to us that when you see a segment of nucleotides containing thymine, that will automatically be DNA. And then if you have a sequence of nucleotides containing uracil, it would be automatically an RNA sequence. All right. And then we have here the second difference. The RNA polymerase can start an RNA chain without a primer, while the DNA polymerase requires a primer in order to start replication. The RNA polymerase is less accurate, and the DNA polymerase will be having high accuracy. Now, we also need to take note of the different types of RNA. So there are 5 types of this, and you can remember this by making use of the mnemonic MRTSS. MRTSS. So what are these? The first one, M, is mRNA or messenger RNA. The second one would be rRNA or ribosomal RNA. The third is tRNA or transfer RNA, while the S would be snRNA, small nuclear, and the other S is SNOR, which would be small nucleolar. Now, of course, you don't only need to memorize the names of these 5 types of RNA, but also know the functions of these rRNA. So we have here the different RNA types of RNA and their functions. So starting with the mRNA, messenger RNA, you know that this would be coding for proteins. This is a template made use by the ribosomes to produce your amino acid sequence or your proteins. The rRNA, ribosomal RNA, would be forming the basic structure of the ribosome and catalyzing the protein synthesis. The tRNA or transfer RNA, by the name itself, transfers. So it will be the one that will be transferring the amino acid and making use of that so that you can produce the amino acid sequence. While the snRNA, the small nuclear RNA, would be functioning in the different nuclear processes like mRNA or splicing inside of the nucleus, while the snORNAs, small nucleolar RNA, will be used to process and modify the ribosomal RNA. So those are the 5 different types of RNAs and their functions. So you need to remember